Thank you. So I'm probably a bit of an imposter because I'm really a quantitative genetics person who just happens to apply things in psychiatric disorders. So this is really a kind of methods talk which segues, I think, hopefully nicely with some of the things that have gone before with a bit of a schizophrenia twist. And one of the things I've realised by listening to the other talks is, in a way, how we live in our own bubbles and see things from our own points of view. And one of things today's presentations have shown me many publications I wasn't aware of. And so with that in mind, I'm going to start with my view of where polygenic risk prediction comes from. And this is actually the first place that I know it was mentioned. It's in 1990 when we were in the RFLP type territory. This was a paper which... So the key thing about polygenic risk score prediction is recognizing that it's genome-wide, that it's leveraging linkage disequilibrium, that it's trying to identify causal variants, maybe thousands of causal variants buried amongst hundreds of thousands of non-causal variants. So this was a paper by Landy and Thompson, and as you might expect, as Eric said, it was in the context of selection. And so then the next key paper is 2001. This is, again, 2001, it was anticipating dense SNP arrays. It talks about polygenic prediction. And the livestock industry actually have taken this methodology on board, have implemented it very quickly, and already by 2016 are presenting results on how the genomic methodology has improved things. And if you're interested, that's a paper published this year where I make a comparison between polygenic risk prediction in humans and livestock, because we can actually learn an awful lot from livestock because big data has been there for a very long time. So it's because of my background in livestock genetics that in about 2006, when everyone else was talking about the Wellcome Trust Case Control Consortium, which is where we kind of benchmark genome-wide association studies, Peter, Mike and I, who all have livestock backgrounds, said, well, there's this great excitement about what's going to be discovered, but in fact our understanding of polygenic disease means that we think that where we're going to get most gain is from risk prediction. So this was before there was data, so this was a simulation study, and again the key thing was what's new is about it being genome-wide. It's about there being, say, 1,000 causal variants amongst hundreds of thousands of non-causal variants, and it's about that estimation of those betas. And the outcome of that study was to say that the value of these predictive SNPs could be reaped long before we understand the causal mechanisms of what we discover in genome-wide association studies. So we took this simulation and applied it as soon as we can to the first data set, and this was actually a schizophrenia data set. So this is the International Schizophrenia Consortium. So Peter, Stuart and I in Brisbane took the data. So this was when there was 3,000 schizophrenia cases. There was one genome-wide significant locus. Everyone was about to abandon genome-wide association studies for schizophrenia. And we said, well, let's look at the data in a different way, and did this out-of-sample prediction, took one cohort out and predicted into it. And we did the analysis, bashed it around as best we could, couldn't get rid of it, handed it over to Sean Porcell, who bashed it around as best he could, and did many, many simulations. And so this is the iconic sort of graph, which maybe you've seen before. So this is where the p-value thresholding method was introduced, where the Plink score command was introduced. And in fact, we'd had terrible trouble trying to publish, well, not terrible trouble, but it was rejected by the American Journal of Human Genetics, because at that time, this sort of methodology was just too far out there. We were very concerned about putting in a genetic risk score, and so we just called it SCORE in this paper. And so what else are the key things? So this also then introduced prediction into a different disorder. Here we've got prediction into bipolar disorder, showing the polygenic link across disorders. What comes next? Okay, so this is now going to the 2014 schizophrenia paper. So this is now taking the same target sample, the MGS data set, and this is then the results presented here in the paper. So this is the same data. You can see the scale is the same. International Schizophrenia Consortium was the discovery sample, 2,600. PGC, Psychiatric Genomics Consortium, dataset 1, 9,320 cases, and this is the published one in 2014 with 32,000 cases. And you can see this increase in out-of-sample risk prediction. And as Montserrat said, it's very important to look at this out-of-sample prediction in different data sets. So this is a bit of a messy graph, but basically each of these is a cohort, and it's showing that we're getting out-of-sample prediction in all of them. Again, it's a bit messy because it's got many of these p-value thresholds. This is the same data presented in a different way with a different evaluation method, a different type of R-squared, and now ranked by the out-of-sample prediction. So you can see, some data sets not as much out-of-sample prediction as others. So one thing that we've thought about is, can we actually improve out-of-sample prediction by leveraging information from correlated data? So we know that schizophrenia, well, we didn't know at the time, but now we know that schizophrenia and bipolar are highly correlated. Can we leverage the information from those different disorders in out-of-sample prediction? And so this is the results here. This is out-of-sample prediction to bipolar disorder using just bipolar disorder and then adding on information from schizophrenia. The different colours are just different methods, but one's single trait and one's bivariate. And similarly here, a single trait for schizophrenia and then adding information from schizophrenia. So a small increase, but another way to look at it is the increase that you get in terms of sample size. So this is the sample size for schizophrenia single trait, and this is the equivalent increase in sample size on this x-axis by adding the correlated trait. So you do get a gain in out-of-sample prediction. So to schizophrenia. So you might say, well, why would you want to predict schizophrenia? We've talked a lot about prevention, and maybe you wouldn't want to be predicting psychiatric traits in a population setting. But there is a great need for predictors in psychiatric disorders because the onset is, as you all know, there's a spike in onset in late teenage and adulthood years, but there's actually a long time to diagnosis. And even the diagnosis, we superimpose this, dichotomous type diagnoses onto quite continuous phenotypes. And so there's a very long prodromal period where you have young people attending, often seeking help, but not having a diagnosis. And so these are papers showing that there's a need for predictors, and they're also saying that the difficulty in psychiatry is we don't actually know what we're trying to predict. We don't have this hard outcome diagnosis for prediction. This is by Tom Insall, former NIMH director. This paper outlining the need for predictors. And this paper from my colleagues in Australia, they, I think, are leading in their ideas about how we should have clinical staging in psychiatry, just as you have in other disorders. And by clinical staging, you can then evaluate young people as they present in youth mental health clinics and triage and decide how to treat based on this kind of more objective staging type of method. So this is now looking at that with real data. So this is my collaborator Ian Hickey in Sydney, and he, together with Pat McGorry, have a cohort of, nearly 1,500 young people, average age 19, who have attended very broad mental health service clinics, including what we call Headspace, which is, we have this kind of walk-in high street mental health clinics. And of those, 209 were invited to participate in a neurobiology phenotyping study. And we had usable DNA from 182 of those, of which 176 were European. And so I generated genome-wide SNP data on these a few years ago, and it's such a small cohort, I didn't know what to do with it. But with the UK Biobank, I've been able to re-look at those data, because the UK Biobank gives us a way to benchmark the risk scores that we can get in that data set. So what I've done is taken the SNPs, which are past QC in this cohort, matched them to SNPs in the UK Biobank, generated polygenic risk scores for schizophrenia in the UK Biobank, standardized those scores, and then could apply the standardization to the scores from our small cohort. And so I've looked at the scores for height, and you can see, that the scores for height are, approximately normal. It's only a small cohort. For BMI, also approximately, distributed about the mean. But you can see the risk scores for these young people presenting this clinic are very much shifted to the right. So then you might say, well, okay, if they're all shifted to the right, well, it doesn't actually get us any further. But there are a few individuals here who have very high scores, which really are way beyond the tail in this normal, UK Biobank distribution. And part of this clinical staging is about identifying ultra-high risk groups when they present at youth mental health clinics. And part of that ultra-high risk identification uses family history. So essentially, these are a set of individuals who have high genetic risk, just as those with family history have high genetic risk. And one of the things about family history is that we know that under a polygenic model, most people don't have a family history. So this is modeling, but it's modeling based on a polygenic model, and this modeling fits real data. So for a disease of frequency 1% in the population, which is like schizophrenia, and of high heritability, like schizophrenia 80%, we know that about 75% of people have no first-degree relatives, 65% have no first, second, or third-degree relatives. And so we are using family history in identifying ultra-high risk, but we could now be using polygenic risk scores to also be identifying ultra-high risk. So polygenic risk scores for schizophrenia have been used in many out-of-sample prediction papers. I'm not going to go through them all. There's a systematic review about them. I've picked out these results because this is prediction into the Danish data set. So as you know, Denmark has got nice electronic health records. The eyesight cohort selected all people who'd had a diagnosis of schizophrenia and took totally random controls. And here you can see that the polygenic risk prediction actually doesn't work as well in Denmark as in other cohorts, and that's partly because it's the idea is it partly is a totally real population data set. But we can see that the risk predictor does separate out based on chronicity, and that's been shown in different ways. But it's actually not that specific because the red line is inpatient for any psychiatric disorder. And that really follows also the known correlation between data sets. But the key thing about these risk predictions is also to understand what's in the discovery sample. That when people have looked at chronicity in the target sample, they need to take account of the fact that our discovery sample has actually been very highly over-ascertained for patients who are treated with clozapine and treatment resistant. And so, understanding what's in that discovery sample is also important. So now I'm going to go through a few methodological things. So it's maybe talking to some of the things that Genevieve was saying about how you evaluate out-of-sample prediction. And with disease, often you're finding that people are using Nagelkerke's R-squared. And the reason they use that is because that's what we used, I think, in that very first paper. And there's a reason that I used that in that very first paper. I think I'm probably responsible for this. We used it because I wasn't that fond of AUC at the time, and it seemed like an R-squared, and an R-squared, we know what R-squareds are like in linear regression, and I think I didn't really understand the properties of R-squared for a disease. So what I've taken here is a predictor which explains 7% of variance in liability, and shown what the Nagelkerke's R-squared is, dependent on different diseases. So this is a disease of frequency 1% in the population, which is like schizophrenia. And it's showing that this would be the Nagelkerke's R-squared if we had 50% cases and controls. And this would be the Nagelkerke's R-squared if we had, whatever it is, 20% cases versus controls. And so it is a statistic which is dependent on the proportion of cases in the sample. And just pointing that out as a way to recognize evaluation. And so one of the things you find people do is that they have their data set and they subset it, and then they look at the Nagelkerke's R-squared. And of course, if the subsets end up with different proportions, then that could bias those interpretations. So this is then looking at the results for schizophrenia again from the 2014 paper, the different methods for evaluating it, and how to interpret them. So the Nagelkerke's R-squared gives a mean value across these data sets of 15%. We then said, well, actually, it's better to put this on the liability scale because that then puts it more on a scale that we interpret heritability and SNP-based heritability. So this is now the out-of-sample heritability attributable for out-of-sample prediction. And that's 7% for that same data. So why didn't we use area under the curve? Well, at that point, I had a bit of a bee in my bonnet about the fact that I felt that genetic risk prediction was being evaluated as if it was could ever explain all variation. And of course, a genetic predictor can only explain the genetic component. And the genetic component is different for different diseases. This is talks again to Montserrat's pictures. But at the time, again, you have to acknowledge these things in the history. That didn't seem like that was being interpreted very well. Now I like area under the curve more, partly because you can then interpret this as the probability that a case ranks higher than a control, and that doesn't have some of the issues with the sampling. This, of course, is another way that we see results presented in these deciles. And so this, again, was the slide from the PGC schizophrenia paper. So this is the 10 deciles without a sample prediction to 3 cohorts. The red one being the Danish cohort, showing the lowest out-of-sample prediction. What I've done here is reanalyze the data. So I've actually got, this is just a reproduction, just based on my own analysis. But this is recognizing, what I'm trying to say here is that these decile plots can seem misleading because, again, they're based on 50% cases and 50% controls. And if you recognize that in a population it's 1% cases, then the odds ratio compared to the first decile is really quite different, 20 versus 9. What else? And so one of the questions that we ask is, and again this links into Montserrat's talk, is what is the maximum out-of-sample prediction we could expect? So back in this very first application, we did lots of simulations. Sean did lots of simulations, and lots of simulations under many different genetic architectures. And the key feature was that we had to simulate a data set where 30% of the variance in liability could be attributed to the SNPs that we measure in order for us to get this out-of-sample prediction of 3% on Nagelkirkers R-squared for our sample size. And so this was basically an empirical way of estimating SNP-based heritability, which we can now do in other ways. So if this is what the SNPs were associated with, then this must be the maximum that we could ever hope to be able to achieve in out-of-sample prediction. So what I've got here is what I think is the most under-recognized equation in polygenic risk prediction. It was first introduced here in this 2008 paper. It's been in many methodological papers, but I feel it gets a bit buried, and it's very important. So this is the equation for variance explained in out-of-sample prediction. It's the square of the correlation. And what these papers have done is said, well, what do you expect this to be? And if you make some assumptions, infinitesimal model assumptions, you can say, you can end up with this equation here. And it makes sense because we've got a numerator and a denominator. If this denominator goes to 0, then we can say that the variance explained in out-of-sample prediction is same as the variance explained by all the SNPs together. So this is the SNP-based heritability. And so if this number is big, then this variance, this out-of-sample variance gets small. So let's look at this more closely. So we've got the sample size of the discovery sample here. So if this sample size gets bigger, then this number is going to get smaller. So we're going to get closer to this, this SNP-based heritability in our out-of-sample prediction with the larger discovery sample sizes. But this is also the key feature, is what is the number of SNPs that this is associated with? And this is actually a very large number because this is recognizing that our SNPs are genome-wide, and it's really the effective number of SNPs. And so this number is actually about 50,000 from a genome-wide association study. So we can plug these numbers into this equation. So using the 50,000 for
Naomi, we're at time.
Okay. Right. So I was just going to go through and tell you how we can try and jiggle this and how the methods, different methods, try and change this around to increase that out-of-sample prediction. And just to say that many people might think that we could get better out-of-sample prediction with whole genome sequence data, but that will increase the out-of-sample, that will increase the SNP-based heritability, but it's also going to increase that M very much, and so you might not actually get a higher out-of-sample prediction. And that's just to say the methods depend on genetic architecture, and that's a visualization. The methods to estimate a polygenic risk prediction depend on genetic architecture, and so the best method is going to be different for different diseases. And that was it.
Can you put your summary slide back up so we can at least read it?
See, I was just advertising my conference here.
I think we can take questions during the discussion section. Yeah, so let's go ahead and move on. And we'll hear about atrial fibrillation from Patrick Eleanor.